Hello, everyone. Our topic for today is about the science of microbiology. Microorganisms are important components of the biosphere. In this lesson, you will understand what microbiology is and what organisms are considered as microorganisms. You'll also learn the different groups of microorganisms and how they are categorized and named. At the end of this lesson, you will be able to describe microbiology as a science, define what microorganisms are, describe the system of classifying and naming microorganisms, and identify groups that make up microbial life. So here is a motivation question to start our discussion. Do you think it is important to study microbiology? So I hope you will be able to answer that at the end of this lesson. So first thing first, we know that microbiology is a science. But what is a science? So science is defined as a systematized body of knowledge based on factual evidence. So these evidences must be based on facts. It must be based on research, not just hearsays, nor those that are uh, just just some gossips that are going around the town or something like that. So it must be based on factual evidences. So the word science comes from the Latin word scientia, meaning to know. Okay, so microbiology is a science. Specifically, microbiology is the study of microorganisms. But what are microorganisms? Microorganisms, or what we call MOs, are organisms that are too small to be seen by the unaided eye. So, what do we mean when we say unaided eye? We are actually referring to unaided eyes are those that without something that help them see. Like as of now, my eyes are aided because I have my eyeglasses. Okay? Or those with using contact lenses, their eyes are aided. Or if you use a magnifying glass. So without these things, you know, an organism that you cannot see without these eye aids, okay, they are considered microorganisms. So the field of microbiology revolves around two interconnected themes. First is on understanding the living world of microscopic organisms, and second is on applying our understanding of microbial life processes for the benefit of humankind and planet Earth. So we will try to elaborate more now of these themes. So as we go with our study of microorganisms, the field of microbiology could be considered basic biological science. This is because microbiology uses tools and develops tools to prove or to investigate the fundamental processes of life. You know. We know that many microorganisms are used as model organisms or systems in order to uh, learn something about a specific group of organisms. So say for example, an E. coli. Now, an E. coli is a bacterial model system for the domain bacteria. So, in order for you to understand a whole wide range of uh, bacteria, you don't need to study all of the species. So, you can use E. coli as a representative organism. So this is because These organisms share many characteristics with cells, that of even multicellular organisms. Say, for example, the yeast, no, Saccharomyces cerevisiae, no, is a model system for multicellular eukaryotic organisms. So you don't need to study any more higher organism in order to understand a fundamental process. Now, if you can do it, no, using the Saccharomyces cerevisiae species. Okay. Now, many microorganisms are used as model systems because uh, basically they're very easy to cultivate in the laboratory. So they can be grown to extreme high densities. Okay. Now, microbiology could also be considered an applied biological science because it is at the center of many important aspects of human and veterinary medicine, in agriculture, and in industry. So, for example, many microorganisms are absolutely essential to soil fertility and domestic animal welfare. Now, there are also many organisms or microorganisms that are involved in large scale industrial processes, such as the production of antibiotics, and human proteins, as well as in many of in food production. Microorganisms are important in the production of beer, in the production of cheese, or other stuff. Okay? So the things that you learn when you study microorganism, you could use that to your advantage in order for you to solve something about the no, about what's happening around us. So, for example, many of my research explores the use of microorganisms in environmental pollution cleanup. So, because we understand that they have the capability to degrade or to absorb huge amount of pollutants, so that we could use that knowledge, so we can use microorganisms in bioremediation. So that's just one application, the use of microorganisms. So as a whole, microorganisms are important because even though they are the smallest forms of life, they constitute the bulk of the biomass of Earth. Even though even if you will combine all the people, in terms of the biomass, that is still not at par with the biomass contribution of all the microorganisms of Earth. And just like what I've said earlier, many microorganisms carry out many necessary chemical reactions for higher organisms. Not just like in soil, in the nitrogen cycle. We know that nitrogen cannot be used directly by plants, but because of nitrogen fixers, the microorganisms that are able to convert nitrogen into ammonia or nitrates, then it can be used directly by plants for their growth. Okay? There are also microorganisms in the gut of ruminants, like in cows, that helps these organisms in digestion. Aside from that, the microorganisms are important in the evolution of oxygen because without microorganisms, we will not be here. No, the the animals only evolved when there were already oxygen present in the atmosphere, and that is possible because of the evolution of these oxygen producing microorganisms, like the cyanobacteria, that photosynthesizes and releases oxygen as the byproduct of photosynthesis. And finally, humans, plants, and animals are intimately tied to microbial activities for the recycling of key nutrients and for degrading organic matter. Just like what I said earlier, just like in nitrogen cycle, in phosphorus cycle, in carbon cycle, microorganisms now play very critical roles. So without microorganism, it will be very difficult for us to survive in the environment. Now, microorganisms, just like any other organisms, are also organized into several uh, groups. Okay, so how do we categorize microorganisms? Now, there are different categories on how we could categorize microorganisms. So the broadest category in classifying organisms is the domain. Okay? So that's the broadest category. Now domains could be further divided into different kingdoms, kingdoms into different phyla, the plural of phylum. Phylum is singular. No? Different phyla into different classes, classes into different orders, orders into different families, families into different genera, no? The plural of genus, and different genera into different species. Okay? So species is the most specific category. Okay? So say for example here, the species is Escherichia coli or E. coli or Escherichia coli. That species belong to the genus Escherichia. That genus Escherichia is one of the genus under the family Enterobacteriaceae. Okay? This family Enterobacteriaceae is one of the families under order Enterobacterialis. Then Enterobacterialis is just one of the many orders of the class Gamma Proteobacteria, which is under phylum Proteobacteria. And phylum Proteobacteria is one of the larger phyla under the domain Bacteria. Now, it's very important to note here that the species name is Escherichia coli. No? The coli there is what we call the specific epithet. So you don't say the genus is Escherichia and the species name is coli. No. The species name is Escherichia coli. Just like humans. What is the species of humans? Homo sapiens. Under the genus Homo. The species name is Homo sapiens. So the sapiens there is the specific epithet. It is not the species name. Okay, I hope you will remember that. So those are the different categories, so the level of the hierarchical classification of different living organisms. Again, this is not just true for microorganisms, but for all the other types of organisms as well. Now, there are three domains of life. Okay, so the domain is the broadest category. So all of the living organisms are divided into three domains. Okay? So what are these different domains? We have the domain archaea, the domain bacteria, and the domain eukarya. So just like what you have learned in your previous lessons, the domain archaea and the domain bacteria, the organisms under these domains are all prokaryotic organisms, meaning they do not have their own nucleus. While the only eukaryotic domain is the domain eukarya, meaning all of the organisms under domain eukarya are eukaryotic, meaning they have their cells have their distinct nucleus. Okay? So the archaeans, the organisms under domain archaea, we call them the archaeans, are found on Earth's extreme aquatic environments. So what are these extreme aquatic environments? So very hot environment, very high pressure, very cold, very acidic, very alkaline. So examples of these are the methanogens, the extreme halophiles, and the thermophiles. So thermophiles are those that prefer very hot environments. Extreme halophiles, those are that prefer very salty environment. Methanogens are methane-producing microorganisms. The domain bacteria, on the other hand, are different from archaeans because if archaeans are specialized in their preference for a specific condition, then bacteria do not have such ability because they can be found anywhere. Now they are the most diverse, meaning there are so many species and they are the most abundant and they are widespread. No, they can be found anywhere. Example are the cyanobacteria, the spirochetes, and the chlamydia. The domain eukarya, on the other hand, as what you said, are composed of eukaryotic organisms. Examples are the protists, the animal-like and the plant-like organisms, but they're not really animals and they're not really plants. Now they belong to the kingdom Protista. Also the kingdom Fungi, the kingdom Mycota, which is like the yields, the molds, and mushrooms. The plants, no, under kingdom Plantae, like the algae, mosses, ferns, conifers, and flowering plants. And the animals under kingdom Animalia, just like sponges, worms, insects, and vertebrates and other invertebrates. Now, how was the three domains established? Before, no, there is really no domain system. No, the biologists uh, classify organisms under five kingdom classification system. But due to the efforts of Mr. Carl Woos, no, he was able to discover an archaea because he studied methanogens and he used a specific marker called the 16S RNA. He extracted this 16S RNA and digested the RNA and ran a two-dimensional electrophoresis. The first is two-dimensional. First, he separated the RNA fragments based on the size, just like normal electrophoresis. And then after that, those fragments are then separated based on their pH requirements, or I'm not really sure. No? But two-dimensional, two factors. And he found out that the methanogens, compared with other species of bacteria, have different patterns, meaning it is a whole new different type of organism. And that's why he discovered the archaea. When he proposed then the three domains of life. Okay. Now, if we know that there are different types of microorganisms, how do we name? Because there are so many of them. How do we classify them? How the first step in classification is also naming, the organisms. So the system for naming organisms is called nomenclature. It is one aspect of systematics, and it is established by Carolus Linnaeus in 1735. This system is called the binomial system. Okay. Binomial, bi meaning two, nomial referring to names, so it's a two-name system. So that constitutes the scientific name, which is done in Latin. So all of the scientific name are in Latin. So the scientific name is a universal name that is given to a specific organism. And the name must be in Latin language. Why Latin? Why not English? Why not Bisaya? Or why not Tagalog? Latin, because Latin is already a dead language, meaning it does not evolve through time. Unlike Tagalog or Bisaya, di ba? Our language evolves. The meaning of one word today may differ 10 years from now. But with Latin, the meaning of the word will be the same because it is already dead language. Will be the same through generations. Okay? And that is called a universal name because everyone around the world understand what is meant. If I would say I am referring to the E. coli, the Escherichia coli, everyone knows that the E. coli is that particular organism, not the Salmonella or the Bacillus, but the E. coli that you're referring to. That's why a naming or giving scientific name using the Latin language is very important. Okay. So since it is binomial, okay, so two names are required. So you must have a genus name and the specific epithet. Just like the species Escherichia coli, the genus is Escherichia and the specific epithet is coli. Okay. Now, when you are writing it, handwritten, no, you must underline, but not uh, continuous. No, the genus is underlined and the specific epithet is underlined. However, when you work with computers or when you type it, no, so you italicize, no, the word. Now, how about if you write it with your hands and then you form like an italicized, no? So it does not work that way. You still need to underline. As long as you written it with your hands, then it should be underlined. Now, after it is mentioned once, no, when you are writing a report or a paragraph, when you have already mentioned it once, then you can just abbreviate it with the initial of the genus followed by the specific epithet. So, say for example, at the first sentence you have already described Escherichia coli is under domain bacteria. Then you have another sentence that says Escherichia coli, so you don't need to write Escherichia coli anymore, but E. coli is blah 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 blah. Okay, so that's how we write scientific names. Now, how do we name them? Scientific names can, among other things, describe an organism, honor a researcher, or identify the habitat of a particular species. So say for example, how is the species Staphylococcus aureus named? So this Staphylococcus aureus is commonly found in human skin. If you are referring to the word Staphylococcus, coccus meaning circular or spherical, staphylo meaning grape-like arrangement of cells. So Staphylococcus, it is referring to the morphology of the bacterial cells. And aureus, because the aureus is Latin for golden. No, aureum is gold. So when you culture this Staphylococcus aureus under specific culture media, it will produce golden colonies. That's why it is called Staphylococcus aureus. E. coli, on the other hand, was named after Theodore Escherick. No? So, Escherichia. And coli reminds us that coli is Latin for colon, or large intestine. So that's how they would name microorganisms. So maybe if you will discover a particular type of species later on, if you want your name to be associated with a particular organism, then you could, but usually it is named in honor of someone who has great contribution to science. Okay. Just like in VSU, there is a particular species of dendrobium, an orchid, Dendrobium melanae, which is named in honor of the university president, Dr. Pasiencia Milano. Okay, so that is one way of naming organisms or microorganisms. Okay. Now, what comprises microorganisms? Now, what organisms belong to microorganisms? So, all of the living organisms, actually, all of the domains have representative microbial organisms. So, bacteria and archaea, of course, they are all microorganisms. They're prokaryotic organisms. But eukarya, yes, there are also microbial eukaryotes. Just like algae and plants, the fungi, and microscopic animals, and protists, which are mostly microscopic. So these the three domains of life, are also microbial life. Okay, so this is the phylogenetic relationship of the three domains of life. So when you go to your cell and molecular biology, you will be introduced into how did these three domains of life originate from the very first two cells that evolved in our planet. This is just actually a a table showing you the major differentiating characteristics of the three domains of life. Okay, basically, since eukaryotes have the nucleus, so they have the nuclear membrane is only present in eukaryotes, but not in bacteria and archaea. Not just the nuclear membrane. We are not talking about the genetic material. Okay, genetic material also found in prokaryotes and bacteria and archaea, but they are not just surrounded with the nuclear membrane. In terms of plastids, you cannot find plastids in bacteria and archaea, only in eukarya. In case of the peptidoglycan cell walls, yes, there are in bacteria, but not in eukarya and archaea because they have different cell wall composition. In terms of the membrane lipids, the archaeans differ from eukarya and bacteria. In terms of the ribosome size, eukaryotes have larger ribosomes. So bacteria, the main bacteria contains an enormous variety of prokaryotes. Now all known disease-causing, what we call the pathogenic bacteria. When you say pathogenic, you're referring to an organism that is capable of causing a disease. So there are pathogenic bacteria, there are non-pathogenic bacteria. And also, these bacteria, microbial organisms, have large variety of morphologies. No? They have different forms and physiologies. There are different functions and processes that are found in this domain. No? So, example are the proteobacteria here, which is one of the largest group of bacteria. You have the gram-positive bacteria and the photosynthetic bacteria. You have also a spirochete. You have here bacteria with unusual morphology. You have here filamentous bacteria. Now, they can live on their own, a single cell organism, but sometimes they form colonies or filaments. Okay. So these are filamentous bacteria. But this one here is not filamentous. It's just a unicellular bacteria. You have also here phototrophic green bacteria, a hypothermic aquifix, and this is a highly radiation-resistant bacterium called Deinococcus. Now, archaeans, on the other hand, although we will be talking about them in detail, there are several phyla actually that exist. Another domain, archaean, just like we've said, they grow at environment, extreme environment, very hot, very cold, very high pressure, very acidic, those types of environment. So example is the extremely halophilic archaea. So this could survive a very high salt concentration. Microbial eukaryotes could be those that belong to the diplomonads, trichomonads, the flagellates, slimodes, the ciliates, and other animals, microscopic. Okay. So there are many representative microbial eukaryotes. Okay. Example, you have here lichens, which is an association of an algae and a fungi. But they are different from molds, okay, and different from mosses as well. These are specialized relationship. We talk like lichens. These are also microbial eukaryotes, the algae, and this one is also an animal, a protozoan, an animal-like proteus. This is fungi, the protein body of fungi. Now, if there are cellular microbial entities, there are also what we call acellular microbial entities. Acellular meaning, if you notice, there is a prefix a, meaning they are devoid, nor there is absence. So these microbial entities are not composed of cells. They do not really have an organismic characteristics, and they are not even considered microorganisms. I mean, they are not really considered organisms, but they are just studied in microbiology because they are important in studying diseases and interactions with the cellular entities. So examples of a cellular microbial entities are the viruses, and those that are subviral entities, meaning they're smaller than the viruses, like the defective viruses, the viroids, and the prions. So viruses are genetic elements that cannot replicate independently of a living cell, which is called the host cell. So viruses, if you take them out of a cell, it will only take few hours or days in order for them to survive. They cannot survive outside the host cell because, again, they do not have that characteristic. They cannot independently live on their own. No? So example here, this is an example of a cell which is infected with SARS-CoV-2, the virus that is causing the COVID-19. If you notice here, these are the viral particles that are found inside this particular cell. So we know that SARS-CoV-2, the viruses that are causing the COVID-19, cannot survive longer outside the host cell. Okay, although they have higher rates of infection compared to other cells, but they cannot survive for long outside, especially when it is very hot. Okay. Now, although they cannot live on their own, they possess their own genetic information, and their genetic information is independent of the host cell's genome. Okay. Viruses rely only on the host cell for energy, metabolic intermediates, and protein synthesis. Viruses are therefore obligate intracellular parasites, meaning they cannot live outside of their host. So they really depend on their host cell for survival. Okay. However, unlike genetic elements such as plasmids that you can find in bacteria, viruses have an extracellular form. Okay. The virus particle that enables them to exist outside the host, and that facilitates transmission from one host to another. Now, to multiply, viruses must enter a cell in which they can replicate. So that process is called infection. So when you say I am infected with the viruses, that means the virus has already entered your system. It has already entered a particular cell in your body. And different types of viruses have different preference or types of cells that they infect to. Although there are other type of cells, or there are other types of viruses that could infect a wide range of cell types, but most of the viruses, they have a specific type of cells that they infect. We call that cell tropism. Just like HIV viruses, they only infect some macrophages and CD4 cells. For others, like the hepatitis virus, they infect liver cells, things like that. Okay. Now, another type of subviral entities are the defective viruses. So, defective viruses cannot infect a host cell alone, and they rely on helper viruses to provide certain functions. Okay. Some of them merely rely on intact helper viruses of the same type to provide desired function. So, these are called the defective viruses. Now, these defective viruses are also called the satellite viruses. So, there is no intact version of the same virus exist. Thus, these satellite viruses, they rely on unrelated viruses known as helpers. Okay? Viroids is another subviral entity, which are infectious RNA molecules that differ from viruses in lacking a capsid. So, because you see, in viruses have the genetic materials that is wrapped around a protein coat or shell. We call that the capsid. But viroids do not have that wrapping. They do not have that capsid. So, as an RNA alone, they are capable of causing the disease. So they have a reasonably stable extracellular form that travels from one host cell to another. They are small, circular, single-stranded RNA molecules that are the smallest known pathogens. They range in size from these nucleotides. Viroids can cause a number of important plant diseases and can have a severe agricultural impact. So if you notice here, now this is an example of a plant, a tomato that is infected with a specific viroid, the PSTV or the potato spindle tuber viroid, and it's a normal, the control one. So you see what happens when this particular viroid has infected the plant. No? Now, as of the present, there are no viroids that are known that infect animals or prokaryotes. I think that is something not to be thankful about. So far, viroids are only found to infect plants. Now, last of those subviral entities that we have presented earlier are the prions. Okay? Prions are infectious agent composed of protein in a misfolded form. They are not considered living organisms, but are misfolded forms of protein molecules that may propagate. Now, how do they reproduce? They transmit a misfolded protein state. So, what happens is that when a pathogenic prion enters a host cell that is expressing not the native prion, the normal protein, it will promote that infective prion will promote the conversion of the normal protein into becoming an abnormal protein, to becoming a prion. That's how they will infect. That's how they will cause the disease. So the pathogenic prion does not subvert host enzymes or genes as a virus does. Rather, it replicates by converting the native prion proteins that already exist in the host cell into a pathogenic form. So if you notice here, this is a normal protein and this is a misfolded form of that protein. They are expressed by the same gene. So as the pathogenic prions accumulate, they form insoluble aggregates in the neural cells. And that leads to disease symptoms that are invariably neurological, and in most cases are due to the destruction of brain or related nervous tissue. So you have here an example now of a neural tissue, okay, with infections of a prion protein. Those, the red stain, Canacilla red stain, those are the prions that are infecting a particular neural cell that has been causing disease. Okay? So in summary, let me give you these questions to wrap up our discussion. So what is microbiology? What are microorganisms? How do we classify and name microorganisms? What are the different cellular microbial life? How do they differ from each other? What are the different cellular microbial entities? So as you review these lessons, try to answer these questions. It will help you review the basic concept of this lesson in order for us to achieve the objectives that we have laid earlier. Okay, thank you very much. And if you have any queries, questions about the topics, you can contact me in this email. You can also directly ask your questions in our Facebook Messenger official group chat. Okay, so thank you very much and see you again in our next discussion.